So welcome everybody to our webinar today. It's a pleasure to have you all here and that you decided to listen and to learn more about our topic, which will be presented by our guests. My name is Jana Seifert. I'm the co-lead of the Human Microbial Genomics Network, and together with Milka Popova and Yan-Fen Zheng, we welcome you today to this webinar. The webinar today will be about the rules and the obligations we have for sharing microorganisms, especially which we are interested in from the rumen microbiome. But we get also much more information. And we found this topic quite interesting for you because we had our last webinar beginning of the year where we talked about new culturomics techniques, and we faced the issue that a lot of people don't know about the obligations and the rules we have to fulfill when we want to share our microorganism. So therefore, we thought it's a good idea to ask someone which is really in touch with this topic. And luckily, in Germany, we have the German Nagoya Protocol HuB. HuB stands for Help and Questions. So, and I asked them to introduce us a bit more to the topic and gives us some background information and some help on this. And I'm really thankful to Melania Munoz Garcia as well as Tatiana Klages, which are here today as our speakers, and that they agreed and willing to present and give us some more insights into these things which we have to consider. A general comment from my side: this webinar is recorded and the video will be available directly through the GRA channel and the website, so the Global Research Alliance. It will not be stored on a local computer. It will go directly to the Zoom cloud. And we will stop the recording after the presentation is finished. That makes you maybe a bit more easygoing and not hesitating to asking questions. So the recording is stopped after the presentation. So don't worry, you can ask your questions afterwards and they are not recorded. To ask your questions, we ask you to submit the questions via the QA function in Zoom, so in the chat, and we will go through the questions then step by step. If you already saw that a question which you agree or you liked, you can use the upvote button to see that we see, okay, this is a popular question, and that we will give the priority to this question. And if you see that we forgot any question and we dismissed them, just raise your hand and let us know. So with this more administrative remarks, I would like to give over the hand to Melania. So it's a pleasure to have you here. And please start your presentation.
Thank you very much, Jana, and thank you to everybody to make some time to learn about the Nagoya Protocol and ABS and a little bit about DSI, or Digital Sequence Information. I will share my presentation. Okay, and let's see. I just would like to start with our disclaimer that everything we will discuss here today will be taken as recommendations or tips, or like the recommendations where to start with the Nagoya Protocol, but it cannot be considered legal advice. So it is important to clarify the differences, and it is important that if you want to have real legal advice, you ask to your institutions. That's very important. We are here to help, but we cannot give legal advice. And then to introduce a bit our project, this is the German Nagoya Protocol HuB project, and we work in the Science Policy Department at the German Collection of Microorganisms in Germany. This is a part of the Leibniz Association. So it is known as DSMZ, Leibniz Institute. And our project works with science policy in both directions, from policy to science. This is support scientists to understand their obligations with international access and benefit sharing agreements, including the Convention of the Biological Diversity, the Nagoya Protocol, and in the near future, other international agreements, and then also from science to policy. So we organize and bring the voice of scientists and the scientific community to these international negotiations in order to advocate for informed decisions that won't hinder research as we know, as we do it. And this project is funded by the Alliance of German Research Organizations in Germany. So today we will talk about access and benefit sharing and the Nagoya Protocol. Then as you are collecting a lot of microorganisms and store them and exchange them. So we will talk about what is the relevance of the Nagoya Protocol for collections holders. And then I will explain a bit about what are the obligations under the European Union ABS legislation or regulation. And this is more related to compliance with the Nagoya Protocol. And then I will give you some tips about where to start if you need to request an ABS permit and how to build your ABS strategy. And as I said, I will include a few slides regarding the DSI or digital sequence information discussion under the Convention of Biological Diversity. So this presentation will be around 1 hour. I know it's a long time, but I try to include some interactive activities in the middle, some videos, and then you don't get tired of just listening to me. So my recommendation is to sit and relax, but pay attention because at the end, we will ask you a few questions. And to start, I would like to know who is in the room, and I think it's also good for you to meet each other. We don't have the time to go for everybody's presentation, but I invite you to scan this QR code with your phones or enter into www.mentimeter.com and include this code. And then you can participate not just in this part of the presentation, like the introduction and ice breaking, but also in the other sections that also will be interactive. So you can use your phone, you can also open a window in your computer, and you can start registering for the interactive activities. So I know there is still a few people connected, but you will always be able to see the code. Well, not always the code, but at least the use code, and you can start with your participation. So the first question, just to warm up, is what is your favorite hobby? So now I see some people interacting. You can include one or more words in the quiz. So we will see more or less which are what you want to do or what you like to do during your free time. So as I see now, most of the people like reading, learning, swimming, running, some violin players, and then, yeah, some sports and exercising. Interesting. So now we will move to the next question. And now I would like to know what is your research about. So what kind of organisms are you studying, and what kind of methods are you applying in your research? What kind of objectives you have, what kind of methods you use? So try to describe this with few words. So gut microbiome, anaerobic fungi, anaerobic organisms in general, zoonotic diseases, some fungi, food mycology. Multiomics, interesting. Microbiome. Genomics, vaccine development. Interesting. Okay, thank you very much. And then I would like to know, what countries do your samples originally come from? Where you usually take your samples, from which countries? This is important for the Nagoya Protocol because, as you will see later, there are some countries that have regulations and there are some countries that decided to do not regulate access to the genetic resources. So we can see here some countries within the European Union: Croatia, France, Germany, the Netherlands, Spain, but also other countries in other places: the UK, Uganda, Colombia, Kenya, Balochistan, Argentina, China, Japan. Okay, very interesting. But it looks like most of the people obtained their samples from Croatia, Kenya, and Germany. Then next question is, in which country is your institution based? Where are you performing this research? And I ask this question because I will explain the compliance regulations for the European Union, but also some other countries could have and should have compliance regulations. So, for example, the UK. So you're doing your research in several countries in the European Union, but also Cameroon, Uganda, Argentina, Japan. Las Bella University, Balo. Okay. Austria. Okay, thank you. And then the last question in this section is, what is your experience with the Nagoya Protocol? You had no idea about what it is before this webinar, or you have heard about it, but you're not sure where to start, or you have heard about it, but you don't have practical experience. You have never requested an ABS permit, or you have some practical experience that you have already requested at least one permit. You have had this interaction with the provider countries and request an ABS permit. Okay, so we can see that the audience is more into the beginners, right? Or they have some knowledge, but they have no practical experience. But then I think this webinar is going to be very useful for you, because we will start from the very beginning, explaining about the Nagoya Protocol and which are the obligations for scientists. Perfect. So let's start. First, I would like to give you a general overview about what is ABS and what is the Nagoya Protocol. So it's not a secret for any of us that we are losing biodiversity in all continents in the world, and it has important consequences for our day-to-day life. And then the United Nations tried to face this problem, and they adopted the Convention on Biological Diversity in 1992 with 3 objectives: the conservation of biological diversity, the sustainable use of this diversity, and also the fair and equitable benefit sharing. Then I will explain a bit more about the 3rd objective. And then in 2014, the Nagoya Protocol entered into force. And the Nagoya Protocol is basically to implement the 3rd objective of the Convention of Biological Diversity, and it is called the Nagoya Protocol on Access to Genetic Resources and the Fair and Equitable Sharing of Benefits arising from their utilization. And it's basically an international framework for access and benefit sharing, and also for compliance. So we will go into the details. But what is ABS? ABS is access and benefit sharing, and access means taking and using biological material for research and development, but also ask for a permit or for permission first. And then benefit sharing is about giving something back. And this something back could be sharing research results, joint research and publications. That means having co-authors in your publications from the country where you obtain the material. You can also offer some training and in-kind support to the local scientists and so on. And then this benefit sharing should contribute to conservation and sustainable use of biodiversity in the country where you obtain the samples. So these are the three pillars of the Nagoya Protocol: access, benefit sharing, and compliance. And for access, each country decides if they establish rules for access their own genetic resources, the genetic resources that are within their jurisdiction. And this is not implemented at the EU level, so each member state can decide if they regulate access to the genetic resources. So for example, Spain, France, Croatia, Belgium, and other countries, Malta, they regulate access to genetic resources, but some countries like Germany or the Netherlands, we don't, or the authorities decided to do not regulate access to the genetic resources. That means you can use the genetic resources from these countries without the need to obtain a permit. Then benefit sharing is something that you negotiate with the provider country. So this is a legal contract where you commit to give some benefits back. And then the compliance, at least within the European Union, this is regulated at the European level. So the EU ABS Regulation 511 established the obligations for all users within the European Union and the obligations to submit the due diligence declaration. So basically, the European Union is ensuring that everybody doing research within the EU comply with the Nagoya Protocol laws in the provider countries and obtain the necessary permits. And why is this important? At the end of the day, access and benefit sharing measures ensure that all research results support provider countries. And this is about giving something back to these provider countries. So this information you generate with your research could be used to make informed decisions on conservation, sustainable use, and management of their own biodiversity. And the second one, I think, is very important, especially for this group. The results you develop could be implemented could be shared to implement innovative solutions for productive activities, including livestock agriculture, and then contribute to sustainable development. So, for example, I think it's very relevant for you, and some kind of benefits you can offer to give back are help countries to reduce and track their livestock emissions, for example, as results of your research. Then the benefit sharing can also help to build capacities for local researchers to solve local problems. So this is also an interaction you could have and you can offer to the provider countries as benefits. And then other kind of monetary and non-monetary benefits that could be more concrete according to your research that you agree with the provider countries. And what is the relevance for ex situ collections? And here we can consider ex situ collections like museums or herbariums or microbial collections, but also within your own research, you also have smaller collections of the microorganisms you are sampling. So just as an example, as I said, I work in the German collection of microorganisms, DSMC, and it houses the most biologically diverse microbial collection worldwide. We have around 84,000 bioresources and around 2,500 new deposits. We receive around 2,500 deposits every year, and we distribute around 43,000 bioresources to a year through or around many countries around the world. So as we as you can see, we move a lot of strains. We receive a lot of deposits, and we also distribute a lot of strains around the world. And that's why DSM must fulfill international obligations for exchanging this biological material. Even though it is important to clarify that the mere storage of biological material without conducting any research is out of the scope of the EU ABS regulation and the Nagoya Protocol. But if you start doing research, you need to comply and you need to obtain the needed permits. So as we had these huge exchanges of materials and we wanted to do the things correctly, with the international obligations, we decided to become a registered collection under the EU regulation. So this is a voluntary measure for compliance, and DSM applied to this, and it was the first registered collection in the European Union. So that means we changed and we updated our catalogs, and we include the necessary information about the Nagoya Protocol. So when you want to submit deposit into the DSM, you also have to bring the ABS documentation and permits. And as you can see in the picture here, so for example, for this strain, we included the Nagoya Protocol restrictions when the sample comes from a country that regulates access to genetic resources. And also, there's something missing here. Yeah, I remember. Okay, and then if there are not restrictions, we can say there are no restrictions for this specific strain. So it was a lot of work. It was a 2.5-year process to update all the information and the catalog, but then we had important outcomes. Our users can easily fulfill their Nagoya Protocol due diligence requirements because they obtain all the Nagoya Protocol relevant information from us. It increases the usability and legal certainty of DSMC collection. All the strains we have here will be compliant with the Nagoya Protocol. And then as we develop important expertise related to the Nagoya Protocol, we start having more and more projects related to, to this. And it gave rise to the Science Policy and Internationalization Department that we have nowadays. Then this is important to mention for all ex situ collections that should compile and transfer the Nagoya Protocol relevant information to third users. So for example, it's a good practice for collections and also for labs and researchers to store Nagoya Protocol relevant information and documentation together with the biological material. So it facilitates future transfer of information. So it is important that when you transfer material and when you receive material, the Nagoya Protocol relevant documents go also with the material. And it is important to mention that it's very difficult for the end user to obtain the Nagoya Protocol relevant information without the collection facilitating it. So if there is a researcher that would like to use the strains you have in your collection, if they cannot receive the Nagoya Protocol information, it will be very difficult that they can obtain these documents from other sources. And then I would like to mention the importance of the relationship of the Nagoya Protocol with the International Code of Nomenclature of Prokaryotes, the ICNP, and specifically related to this description of new species. So there are sometimes incompatibilities. For example, the code requests that every single new species is deposited in 2 different collections in 2 different countries without restrictions. So then the type strains can be distributed and transferable to third parties to do more research on them. And then if the ABS permit has restrictions, for example, to request a permit before the distribution from the collections may not accept your deposit. So it is important if you are planning to describe new species, you make sure that you can deposit this species in a collection, and this deposit would be with no restrictions for further transfers. for basic research. So this is for collections, but if you are actually performing research, the Nagoya Protocol world has two important parts. First part is the obligations according to the provider country's legislation. So you need to apply for the permits if you are within the scope of the national laws. And then once you have your permits, you may have also obligations under the EU regulation if you are performing your research within the European Union. This is a very helpful infographic that you can access in our website. I will share the presentation later, and then you can use the links. And then you can click in different parts of the infographic and expand the information you receive. But then it's very clear from this that people working within the European Union have to check the two things, the obligations in the provider countries and the obligations in the European Union. And the scope of these two sets of rules could be different. So that's why it's important to check both. And what are my obligations under the EU ABS regulation? To show this, I will start with a short video.
Are you an academic researcher working in the life sciences in Germany? Do you work with non-human biological material like bacteria, fungi, plants, and animals? Does your research material originally come from outside Germany? If so, you might have to comply with Access and Benefit Sharing Rules, or ABS for short. Let's take a closer look. Firstly, you need to comply with laws in the provider country where the material comes from. That means asking the provider for permission to do your research with the material and sharing benefits from your research with them. Secondly, you might also have additional compliance or so-called due diligence obligations in Germany. What happens if you don't follow these rules? It can lead to negative press and reputation damage to you and your institution. It may also make it impossible to publish your research or lead to the retraction of your scientific papers. And there is more. The German Federal Agency for Nature Conservation is checking that scientists and institutions in Germany are doing their research in compliance with ABS rules. If not, it can stop you from continuing your research, prevent you from publishing your results, confiscate your research material, and give you a fine of up to 50,000 euros. So know what your ABS obligations are and follow the rules. It ensures that benefit sharing happens. It builds trust with providers of material, and it facilitates scientific collaboration. ABS compliance supports your science. For more information, check out www.nagoyaprotocol-hub.de.
Yeah, so as you can see from the video, this is serious. And then you need to know which are your legal obligations with the provider countries. And then you have to check which are the legal obligations within the European Union. And if you are doing your research outside the European Union, you have to check your obligations within the compliance legislation in your country. Today, I will focus a bit on the EU regulation on compliance, but this is very useful for the ones doing research here in the European Union, but it's also interesting for the others because in any moment, all countries should be developing this kind of compliance regulation. So whether you have legal obligations with the EU is determined by this regulation number 511 from 2014. And there are 4 important elements you have to consider when you are analyzing if you are within the scope of the regulation, and it is the type of material you're using, and keeping in mind that also associated traditional knowledge may be within the scope of the EU regulation. Then the type of research you're doing, then when the material was originally obtained and where the material was originally obtained or where the material come from. So within the German Nagoya Protocol HuB, project, we prepared the following 4 questions, and this will help you to find out if you are within the scope of the EU ABS regulation. So first question is to check the material scope. So is if your samples contain DNA, RNA, or derivatives. If your answer is yes, you may be within the scope of the EU regulation. And what do you mean with this biological material we are mentioning a lot? It is any material that contains DNA or RNA. It could be dead or alive, because sometimes we think that if we are not obtaining living organisms or parts of organisms, it is not within the scope. But if you go to a herbarium and you obtain a sample from there, it may be also within the scope, because you can extract DNA from there. Or if you are doing archaeological research and then you are extracting accent DNA, you may be also within the scope. So this biological material is any material from any living organism that is not human. So humans are outside of the scope that contain functional units of heredity. This excludes human DNA, but human pathogens and human microbiome are still within the scope of the Nagoya Protocol and the EU. It also excludes plant genetic resources listed in the annex of the International Treaty of Plant Genetic Resources for Food and Agriculture. And it also excludes influenza strains under the Pandemic Influenza Preparedness Framework, or the PIP framework. And it is important that to be excluded of these 2 international instruments, it must be within the scope of these agreements. So for example, if you are doing research on rice, and rice, you have to check if rice is within the annex of the plant treaty, and yes, it is. And then if you are not doing research for food and agriculture, but you are doing research to develop a cosmetic, so it is not within the scope of the plant treaty, but now is within the scope of the Nagoya Protocol. So just organisms listed, or just crops listed in the annex that would be used for food and agriculture, are excluded from the Nagoya Protocol. And the same from the PIP framework. If you're not using influenza strains, I mean, if your strains are other kind of microorganisms, then you are out of the scope of the PIP framework and within the scope of the Nagoya Protocol. But it's not just a material that contain DNA and RNA, but also containing derivatives. And these derivatives is maybe not a common word for biologists or people working with a natural or biological material, but it basically means any naturally occurring biochemical compound resulting from the genetic expression or resulting from the metabolism. So it includes all proteins, including enzymes, lipids, organic compounds like essential oils or resins and other products of metabolism. So honey, milk, all these products of metabolism are considered derivatives. And they are within the scope of the EU regulation when continuity applies. So, for example, if you obtain together the derivative with the genetic resource, for example, if you obtain a plant from the supermarket that you know that comes from Spain, and then you extract its enzymes from this plant to study them, then you are within the scope because you are obtaining the plant and you are extracting the derivative. So you obtain both together. But if you buy enzymes from a company, you are obtaining just the enzymes. There is no continuity, and this is out of the scope of the EU regulation. Then the most tricky part is the utilization scope. So you need to ask yourself the following question. Are you doing research on the genetic or biochemical composition of the material? Yes or not? So you answer yes, for example, when you do research on a specific genetic or biochemical properties, when you study gene function, gene expression, when you are performing gene modification or genome editing, when you are doing breeding based on traits that are associated to genes, etc. So this is not a comprehensive list, but if you are doing some of these things, you may be within the scope of the EU regulation. On the other hand, if you are not doing genetic or biochemical studies on the genetic or biochemical composition, then you are out of the scope. For example, just the mere storing of biological material in a collection without doing any research is out of the scope of the EU. The mere culturing of organisms in vitro or in the field is out of the scope. Taxonomic identification, for example, just using 16S or 18S to identify some microorganisms is out of the scope. And also the morphological or anatomical characterization is out of the scope of the EU regulation, but take into consideration that national legislation from the provider countries may be broader. So, for example, most countries regulate the taxonomic identification within their ABS laws. So you will be out of the scope of the EU regulation, but you still have obligations under the national legislations. And here we will start another interactive practice just to follow up a bit with what utilization of genetic resources and derivatives mean. So I invite you to join again the Mentimeter and give us a thumb up if you are connected. And you are now enabled to participate in the questions. So the first case is environmental DNA metabarcode analysis of water samples to discover the number of fish species present in the water. So the scientists take water samples from river to discover the number of different fish species that are in that river. The samples contain DNA released into the water by these organisms, and to obtain a biodiversity inventory, the DNA is purified from the water samples. Then DNA markers are sequenced, and then the new sequence then they use the new sequence to taxonomically identify the fish in the water. So according to this description, is this research within the scope of the EU ABS regulation? And then you can answer yes or no. So for the first case, for the environmental DNA metabarcode analysis of water samples to discover the numbers of fish species present in the water, is this out of the scope of the EU or is within the scope of the EU? This is out of the scope of the EU. Why? This is out of the scope of the EU because you are not doing gene expression, you are not doing modification of genes, you are not doing research on the composition of the genetic material. You are just identifying the samples. So it doesn't count as utilization within the EU regulation. It's just a comparison of sequences that you need to obtain the ID or the species from the fish from the water. So this is not considered within the scope of the EU regulation. Yeah, so here the explanation. The function of genes is not investigated because only the sequence is used and the functions are not studied or considered. Such inventory studies do not constitute utilization under the EU regulation. But we have more cases, no worries. Then case number 2 is the storage of pathogens pending a decision on their use in a vaccine. So the researchers collect and isolate different pathogens from hosts in various countries and as part of a global surveillance system and consider from epidemiological analysis to be a potential public health threat. So they collect a lot of pathogens for many countries. Then you have the initial analysis, and this initial analysis do not make it clear which, if any, of the isolates will be needed for a vaccine development. However, the threat is considered sufficient sufficiently great that the preparation of vaccines and diagnosis is requested by the WHO at some point by individual governments around the world. So you obtain all these risky pathogens and you store them, waiting for a call from to develop a vaccine. So these pathogens are collected and stored and ready in an already existing collection, as well as exchanged with other collections. Is this research within, or is this case within the scope of the EU regulation? So the question is if storage of pathogens pending a decision on their use in a vaccine is within the scope of the EU regulation or out of the scope of the EU regulation. At this point, it is out of the scope of the EU regulation. Why? Because you are just storing the pathogens. So at this point, building a collection with the aim of using these strains in the future is not considered utilization with the EU. But if at some specific point there is a call and you need to develop these vaccines, at that point, it is within the scope. So at that point, you will need the ABS permits. Because at that point in the vaccine developing process, you will need to study the biochemical or genetic composition of those bacteria. Okay, and then we continue with the other two questions to check if you are within the scope of the EU regulation. First, the third question is to check the temporal scope, and this is related to when the material was originally collected. So if it was originally collected on or after 12 October 2014, this is within the scope. And why this date is so important? Because first is the date that the Nagoya Protocol entered into force. And second, because this is the date when the EU ABS regulation entered into force. So it doesn't matter if you collected the samples in the field or if you obtained the samples from a collaborator or from an ex situ collection or from a shop, you always need to check when this material was originally collected. So if you obtain this from a collaborator, you have to ask your collaborator when and where did they collect the material and check if you are within the scope or not. And then the last question is to check the geographical scope. So you have to ask yourself if the provider country, I mean the country where you obtain the samples, is a Nagoya Protocol party, and it has ABS measures in place at the time of access. If you answer yes, so you are within the scope of the EU ABS regulation. To obtain this information, you have several sources, so you can check the ABS clearinghouse, and you can access in the link, and then you can, then the ABS clearinghouse is the official source of information where you can check each country profile and check if they are within, if they are party and if they have legislation. The only problem with the clearinghouse is that the clearinghouse is not always up to date. So for checking if the country is party or not, you can fully trust on the clearinghouse, but for the question if they have ABS measures, if they don't have anything published in the country profile, that could mean that they don't have the legislation, but it also could mean that they haven't published their legislation in the ABS clearinghouse. So when you have a country with zero ABS measures in place, our recommendation is to always send an email to the ABS national focal point or the competent authority to double check if really they don't have access regulation. So if you answer yes to all the questions, if the material contains DNA, RNA, and derivatives, and if the research investigates the genetic or biochemical composition, and if the material was obtained on or after 12th of October 2014, and the country is party to the Nagoya Protocol and have ABS measures, then your project is within the scope of the EU ABS regulation. And always, as I said, keep in mind that ABS may apply to you even if the EU regulation does not. That's why we have this figure with the circle. So the EU ABS regulation has a specific scope that we just described it, but then the Nagoya Protocol is even bigger because the scope of the national legislation may be different and could include more things. And then the ABS circle is even bigger because there are countries that are not party to the Nagoya Protocol that could have ABS measures. So non-Nagoya Protocol parties also may regulate access to the genetic resources, and it is important for you to check. And which are the obligations if your project is within the scope? Then the EU ABS regulation established that you must comply with the ABS measures in the co-provider countries. That means you have to seek, keep, and transfer ABS information to subsequent users. So you have to seek or check or obtain your ABS permits. Then you have to keep all this information and documentation for 20 years after the end of your project. That means after the publication of results. And then if you are transferring material to your colleagues, you need to transfer the Nagoya Protocol documents together with the material. And it is very important to check if the permit allows you to transfer the material and allows your partner to use the material for other things that are not included in the permit. So usually the ABS permits are just for specific users and for a specific project. If you want to transfer this material to others, probably these other people need to also request their own ABS permits. And after doing this, you need to exercise the due diligence declaration. That means going to the declare portal and declare that you obtained your permits and submit the permits into the form. So you are enabled to use the material for research in accordance to your permits. So that's why it's very important to read your permits with detail. And if you don't have sufficient information, our recommendation is always discontinue utilization until you have all the need permits. Then under the EU regulation, there are procedures called compliance checks or users checks. In Germany, the German Federal Agency for Nature Conservation, or BFN, is the ABS authority in Germany, and they perform these user checks or compliance checks. So they are basically checking institutions within Germany and check if they are complying with the legislation of the provider countries. So you have to answer a questionnaire. You have to provide all related documents, the ABS permits and so on. Then you have to allow access to your premises if they want to visit you, and sometimes they also request samples. So it is important that all users of genetic resources support these compliance checks. And this is happening in Germany, but we need to know more about compliance measures in other countries and be aware, for example, who are the competent authorities in the countries where you are performing your research, which are the processes and which are the consequences of not compliance in your country. And continue talking about Germany, there are important implications of non-compliance. For example, as you check in the video, fines, orders to stop research, confiscation of material, publication embargo or retractions, but also you can be sanctioned by the provider country with fines and also jail terms. And you will have also non-regulatory consequences, for example, the loss of reputation of you as a scientist, but also your scientific or research institution can lose reputation. You can lose trust in the provider countries, and you could be included in a blacklist in these provider countries. So they won't grant you a permit anymore. So these are some publications in very well recognized journals, scientific journals, but they basically put it on the spot researchers that haven't complied with the legislation, and they are now in troubles. But where to start? I think now you have obtained a lot of information. What is the Nagoya Protocol? What is access of benefit sharing? What is considered within the EU regulation and what you have to do, which are your obligations. But then where to start? So the German Nagoya Protocol HuB created this checklist that help researchers that have never requested an ABS permit before. And as we saw in the quiz at the beginning, this is the case of all of you. So this is very clear guidance of what to do before you request a permit. First, you need to be very well informed. You can check different sources of information, including the ABS clearinghouse, but also talk with your partners and talk with the authorities. And you can also ask questions to the GMP HuB to be very well informed about what the provider countries are requesting. Then you don't have to reinvent the wheel. There are some model agreements. You can ask for model agreements in the provider countries or from your partners, or your institution can develop model agreements. And then it's important you take care of the legal aspects. Who is signing the documents in your institutions, which other permits are needed besides the ABS permits, and which other agreements are needed, for example, an NTA with partners and so on. Then the second part of the checklist is define your research needs, which are the red lines in any benefit sharing negotiation. So you must be sure that all you need is included in the permit. What material you need to sample, if traditional knowledge is included, which methods are you allowed to use with the permit. That means being sure that all the methods are included. And then just any restriction on the results. If you need to deposit material in a collection, be sure that this is included in the permit. If you need to deposit sequences in open access databases, also be sure that this is included in the permit. And as I mentioned at the beginning, if you're describing new species, you need to check if you are allowed to deposit into recognized or in collections with no restrictions to transfer to third parties. And also check that all your collaborations are included in the permit, because if you need to share samples within your partners, you should include your partners in the permit too. Then the third part of the checkline is really think about what kind of benefits you can actually share, because remember that this benefit sharing is included in a legal document. This is a legal contract, and you are forced by this contract to actually comply with the benefit sharing. So you need to check if all the benefits you are offering are consistent with your grant agreement or with your company policies. Define who is receiving the benefits. This should be very clear. If you are giving the benefits to the national authorities or to the local communities or local researchers or in a specific sector within that country, for example, to a specific group of farmers. And then you need to be clear about the timing and scope of the benefit sharing. And obviously you need to report back to the authorities and keep a record. So the most important message here is do not make promises your institution can't keep. And then the last part of the guide is exploring options for future research. So it is important to think for the future, and then you can reduce the burden of asking for extensions. And is if you will need to reuse the material for other projects, or if you can ask the permit for several project objectives. Then you can ask if you can request a modification or an addition to your permit, or if you need a really new permit. And also think about if you will need to transfer the material to other research or institutions in the future. So it is time for the last few questions for you, and then we will go very briefly through the DSI discussions. So I invite you again to go into the Mentimeter with the code or with the QR code, and we will check your understanding of the previous presentations. So we have, I see few people joining. I will invite everybody in the presentation to join, in the room to join. And let's start with the quiz. And for this part of the quiz, if you answer fast, you will obtain more points. So also speed counts. So is true or false that all parties to the Nagoya Protocol regulate access to the generic resources? No, not all parties regulate access. For example, we mentioned that establishing rules to access to the generic resources depends of each country. So for example, Germany decided to do not regulate access, as other countries within the European Union, like the Netherlands and the UK, for example, they also decided to do not regulate access to the generic resources. So this is false. Next question. I am conducting research on material from a country that is not party to the Nagoya Protocol. Then I do not have ABS obligations. Is this true or false? Yes, this is false, because even if the country is not party to the Nagoya Protocol, this country could have ABS legislation. So for example, Colombia is not party to the Nagoya Protocol, but they have ABS legislation and you must follow this legislation. This question is, my research will not produce a commercial product and will not generate profit. I have no benefits to share, and the ABS and the Nagoya Protocol do not apply to me. Is this true or false? Correct. This is false, because the Nagoya Protocol also applies to non-commercial research. So even though you don't generate profit, you can share non-monetary benefits like the research results and training and co-authorship. And this is also within the scope of the Nagoya Protocol. Very good. Then the last question of the day. A colleague in Switzerland regularly sends me organisms which originally come from Thailand. So which country do I have to check for the ABS laws? Switzerland or Thailand? Right. Most of you have the correct answer. As the samples originally come from Thailand, Thailand is considered the provider country. So you must follow the access regulation from the provider country, that is Thailand. Okay, so now we will see who is the winner of the quiz. So we can see that this Casper was the one with more questions correct and also the faster to answer the question. So he's or she is the winner of the quiz. Congratulations. And then I will close in the next few minutes with an introduction to the digital sequence information that we are having under the Convention of Biological Diversity. and why it is relevant for you. So besides the Nagoya Protocol, there are new challenges that are arising from international negotiations on digital sequence information, or DSI. And this DSI is a policy term that refers broadly to genomic sequence data and other related digital data. For example, sequences of DNA, sequences of RNA, sequences of proteins, and also the structures of metabolism. So there is the establishment of a future mechanism for benefit sharing from the use of these sequences, and this put at risk some current condition of open access to large biological databases. So this is all about using sequences that are publicly available in open access databases. So the national authorities heard that scientists don't need to go through the headache of Nagoya, so they don't need to really ask for an ABS permit if they limit themselves to use the sequences in open access databases. So there is a political assumption that open access databases giving all this information openly, and the new methods of synthetic biology will allow to generate commercial products that could be in the market without the need of an ABS permit. So this is producing profit or monetary benefits that are not going back to the provider country. That's why we say this is a loophole in the, or is considered a loophole in the Nagoya Protocol. So there is a call for benefit sharing from the use of these sequences. The problem is that you cannot request an ABS permit per each sequence you download or use from open access databases, because those are thousands of hundreds of sequences that we can use in just one analysis with the sequences. So we really need to develop or to invent a new mechanism to ensure benefit sharing that decouples access. That means we keep open access to sequences from benefit sharing. That means we create a method for sharing the benefits that is not related to the access of the sequences. Then it was and is very important the participation of the scientific community in these international negotiations to really make the point how scientific research work and how some decisions in these international conventions could negatively impact the research. So the biggest dilemma is how we can ensure benefit sharing from open sequence data without damaging or harming open access. So there is the DSI Scientific Network, and the DSI Scientific Network is a group of researchers from all around the world. We have nowadays around 90 members from all the continents, and we start working together to bring the message from the scientific community to the different convention of the parties of the Convention of Biological Diversity, where all these decisions are made. So the idea is to share that scientific community supports benefit sharing, but we cannot ask for permits for individual sequences. So we need to keep open access to DSI or digital sequence information databases, and we share the benefits through multilateral approach. So the users share the benefits to just one mechanism, and the mechanism is in charge to distribute the benefits to the countries. And the important point is to keep it simple in order to do not include or increase regulatory complexity and costs of research, and make it compatible with other international agreements, because then we have the BBNJ agreement for biodiversity, marine biodiversity beyond the national jurisdiction, and we have the Plant Treaty, and we have the WHO, and we have other international agreements that every single fora starts developing new legislation, then we will finish with a very complex way of compliance for researchers. So if you want to go deeper in this multilateral approach for benefits sharing, I recommend you to read the Nature Communication paper called Multilateral Benefit Sharing from DSI. Will support both science and biological conservation that was produced by the DSI Scientific Network members. Then when we had the COP in 2022, the final decision said, well, we will go with a multilateral mechanism and a fund for sharing monetary benefits. And this is a very important win for the scientific community because we don't have to access, or we don't have to request individual permits to be able to use the sequences. And then in the COP 15, they decided the principles that this mechanism should follow. And as you can see here in orange, this is an important win for researchers too, that the mechanism should not hinder research and innovation, should be consistent with open access to data, and should be mutually supportive of other access and benefit sharing instruments. So the request that the scientific community made at this point was considered into the decision. And then after COP15, the most important discussion was how this multilateral mechanism would work. And this is what was negotiated during COP16 4 weeks ago in Cali. So they discussed and negotiated the modalities for the multilateral mechanism and the fund. So basically discussed the contributions to the fund, who will contribute, how much, if this is by product or this is by sectors, and how. And then they also discuss the allocations of the fund, if they will be allocated by projects or direct allocation, to whom, to developing countries, developed countries, to the indigenous people, and what kind of activities should the fund support. And other issues discussed was important for the research community was, or were the role of DSI databases, the non-monetary benefit sharing. That means that benefits that are not shared in cash, but in kind. For example, open data, open publications, and the databases, and so on. And also the governance of the mechanism and the fund. And it was a very intense discussions in Cali for COP16. And also, again, the DSI Scientific Network sent the message to the COP. So the important thing is continue the database practice we have nowadays that enable and support open access. Then be sure that commercial users should not have obligations to pay to the fund. Then allocate also resources for capacity building to generate and use DSI. Develop a framework for non-monetary benefit sharing. This is consider that all the scientific community is supporting the databases with all the sequences there, because obviously the private sector do not upload their sequences in the databases. Then keep it multilateral and avoid bilateral process to reduce complexity, and also create a mechanism that is compatible with other international ABS instruments. So all of these will support innovation, research and development for conservation and sustainable use of biodiversity, and will also support the global biodiversity framework. And if you are interested, you can scan this QR code and you can read the DSI Scientific Network position paper before COP16. And then what was decided? The COP16 established the modalities for the multilateral mechanism. That means the creation of the mechanism will ensure flow of resources that will support biodiversity and conservation. And it builds a system that incentivizes research and sharing of data. This is super important for the research community. Then the mechanism will cover all genetic data that is publicly available, so we can continue using the sequences as we are used for. And then the decision includes clarity about which users have which obligations when we talk about contributing money to the fund or contributing with open data, open publications, and so on. If you're interested, you can also check this link. It's almost the final decision, because at the end of the plenary, there are some few changes, but you can read, this is the available document we have nowadays. Then it is important to mention that the final COP decision on DSI also reflects many of the scientific community recommendations. So no commercial users are not expected to contribute money to the fund. This is a big win for us. So just big companies are for-profit commercial users will contribute to the fund. And this is very good because they recognize that researchers are contributing with open access data, with open publications, and with information to the countries. Then the databases should operate in a way that is consistent with open access. So open access is recognized as important and protected and supported. And then non-monetary benefits and DSI capacity building is cited as a focus area. So there are highlights that capacity building is needed to all researchers have similar capacities on using the DSI that is publicly available. So here you have a picture of this DSI Scientific Network delegation. We have an important representation in the COP, and in the QR code and the link, you can obtain the post-COP statement for the DSI Scientific Network. And there are still a few things that must be further discussed in the next years before the next COP. And it is the role of databases in supporting transparency on the use of DSI, how non-monetary benefits should be managed within the mechanism, and then the impact of new technological advances such as artificial intelligence on the benefit sharing system. So I think it took a bit longer than I expected, but I hope the information is useful for you. Here you have the contact information. You can visit our website. It has a lot of material that could be interesting for you and that is easily explained things related to the Nagoya Protocol. And yeah, thank you very much for your